The xenobiotics are attacking Earth.
It really does. But the etymology is actually quite literal and very grounded.
Okay.
It comes from the Greek xenos, meaning foreign, and bios, meaning life.
Ah, okay. Stranger to life.
So a xenobiotic is literally a compound that is a stranger to life. It's inside you, but it doesn't belong to you.
Right.
It has no nutritional value, no physiological function in your normal metabolic pathways. It's just there.
And the body has to figure out what to do with it.
Exactly. And the list of these strangers in the source text was massive. It's not just the obvious stuff like industrial pollution or, standing next to a smokestack.
Yeah, that's what I thought at first.
Right. I think when most people hear toxins, they picture like nuclear waste or a vat of green sludge. But the text categorizes them much more broadly. We have drugs, which we invite in voluntarily to treat ailments.
Of course.
We have chemical carcinogens. We have environmental pollutants like PCBs, polychlorinated biphenyls, and insecticides.
But the one category that really stopped me in my tracks was the plant compounds.
Ah, yes.
Because we are trained to think natural equals safe. We think it's from the earth, it must be good for me.
That is probably the single biggest misconception in all of toxicology.
Really?
Natural does not mean biochemically neutral. You have to remember, plants can't run away. They can't fight with claws or teeth.
Sure.
So for millions of years, they have engaged in chemical warfare. They produce these complex molecules specifically designed to mess with the biology of the insects and animals that try to eat them.
So that kale salad is actually a chemical weapon.
In a sense, to your liver, the compounds in that salad are just as foreign and potentially just as difficult to process as a pharmaceutical drug. And that brings us to the mission of this deep dive. We aren't just listing chemicals. We are going to do a comprehensive line-by-line analysis of the machinery, the absolute marvel of engineering inside the liver that allows us to survive this chemical onslaught.
And this isn't just an academic exercise. I mean, the text makes a point that this specific topic is the bedrock of modern medicine.
It is the absolute foundation of three massive fields. It's the foundation of pharmacology, how drugs work, and how we dose them.
Okay.
It's the foundation of therapeutics, how we actually cure people. And it's the foundation of toxicology, understanding what kills us.
There's also this really interesting note in the intro about the future. The text mentions transgenic organisms. It feels like we're pivoting from how our bodies work to how we might, hack nature.
Yes, this is where the science gets really, really exciting because we are beginning to map and understand the genes that encode these metabolizing enzymes. Scientists are now looking at developing microorganisms or even plants that are transgenic, meaning they've been modified to carry specific genes that can process pollutants.
So effectively, we could engineer a bacteria that eats oil spills, or a plant that sucks toxins out of the soil at a Superfund site.
Exactly. Or even using them for the biosynthesis of drugs. If we understand how nature metabolizes chemicals, we can engineer organisms to do the hard work for us.
That's incredible.
But to get there, we have to understand what's happening inside our own bodies first.
Okay, let's go there. Let's go to the liver. I picture the liver as this massive chemical processing plant, steaming and hissing, trying to sort through everything we ingest.
A good visual.
What is the general strategy here?
The general goal is pretty simple. Detoxification.
Okay.
But we need to understand the physics of the problem first. Most of these xenobiotics, drugs, pollutants, plant toxins, they all share a common trait. They are lipophilic.
Lipophilic, meaning fat-loving.
Correct. They dissolve in fat, not water. That property allows them to slide right through your cell membranes and get absorbed into your body.
Okay, so that's how they get in.
But that same property makes them incredibly difficult to get rid of.
Because you can't just pee out a ball of grease, right?
Precisely. Urine is water, bile is watery. To excrete something, you need to make it water-soluble or polar.
Got it.
If you can't make it polar, it just stays in your fat tissue, accumulating forever. So the liver has developed a two-phase strategy to take these greasy, fat-soluble strangers and turn them into something that dissolves in water so we can flush them out.
Phase 1 and Phase 2.
Right. But before we break those down, I need to drop a bomb on you.
Oh.
I mentioned that the goal is detoxification, but the text highlights a crucial, very counterintuitive fact. Metabolism doesn't always make things safer.
Wait, what? I thought the whole point was to protect us.
That's the evolutionary goal, yes, but chemically. Sometimes this process takes a compound that is relatively inert, completely harmless, and converts it into a biologically active one. We call this bioactivation.
So the liver actually weaponizes the chemical.
In a way, in pharmacology, we use this on purpose. We design prodrugs.
Roll.
These are drugs that don't work in the pill form. They are inactive. You swallow them, and your liver performs phase 1 metabolism on them, and boom! Now they are active drugs that can treat your condition.
That's the good scenario. What's the bad scenario?
The bad scenario is when the liver takes a harmless environmental chemical and metabolizes it into a carcinogen or a mutagen.
Oh.
The liver is just trying to process it to get rid of it, but in that intermediate step, it creates something that damages your DNA. That is the dark side of xenobiotic metabolism.
That is terrifying. It's like the bomb squad accidentally arming the bomb while trying to defuse it.
That is a perfect analogy, and we are going to see exactly how that happens when we look at the mechanism.
Okay, let's look at the mechanics of the bomb squad. Phase 1, what is happening here?
Phase 1 is all about functionalization.
Functionalization.
The body needs to get a handle on the molecule. Imagine a smooth, greasy ball. You can't grab it.
Right.
Phase 1 is about drilling a handle into that ball. Chemically, the main reaction is hydroxylation.
Hydroxylation, that means adding a hydroxyl group, an OH group.
Correct. You're attaching a little chemical hook. The main player here, the star of the show, really, is a class of enzymes called monocogenases, also known as cytochromes P450.
We are definitely going to talk a lot about P450 later, but generally, what does it do in phase 1?
It introduces a reactive group. It could be hydroxylation. It could be deamination, which is removing nitrogen, dehalogenation, removing halogens like chlorine or epoxidation.
A lot of options.
But the result is generally the same. The compound becomes more reactive.
And more reactive means potentially more toxic.
Temporarily, yes. That's why phase 1 is risky. You are making the molecule chemically sticky so that phase 2 can happen. You are stripping the insulation off the wire. It's necessary, but it's dangerous if you stop there.
Okay, so phase 1 puts the handle on the suitcase or strips the wire. What is phase 2?
Phase 2 is conjugation. This is where we attach a massive water-soluble tag to that reactive handle we just created.
What kind of tags are we talking about? What's it made of?
The body uses what it has lying around in the pantry, so to speak. Glucuronic acid, sulfate, acetate, glutathione, various amino acids. These are all molecules that love water.
So phase 1 creates the hook. Phase 2 hangs a giant wet sponge on the hook.
Exactly. And once that conjugation happens, the molecule is now a polar compound. It dissolves in water.
And then it's out.
Now the kidney can filter it into urine, or the liver can pump it into bile, and it leaves the body. That is the end game.
So ideally, ingest weird chemical bile. Phase 1 makes it reactive. Phase 2 makes it water-soluble excrete.
Ideally. But as we'll see, the devil is very much in the details.
Let's talk about the hero, or sometimes the villain, of this story. Section 2, the star player, cytochrome P450.
Ah, yes. The P450 superfamily. This is where the biochemistry gets really beautiful and really complex.
First off, let's address the name, cytochrome P450. It sounds like a robot from Star Wars. C-3PO meet P450.
It does. It has that vibe. But the name is actually a history lesson. It tells you exactly how they found it. Back in the mid-20th century, scientists were studying liver cells. They would, grind them up and spin them in a centrifuge. When you do that, the endoplasmic reticulum, which is this vast network of membranes inside the cell, shatters.
And what happens to the pieces? They just dissolve.
No, the fragments actually reform into these tiny little bubbles called microsomes.
Microsomes.
These aren't natural organelles, they're artifacts of the grinding process. But inside these microsomes, they found a pigment.
A pigment, like a color.
A cytochrome, which literally means cellular color. They noticed that if they chemically reduced this enzyme, meaning they added electrons to it, and then exposed it to carbon monoxide gas.
Which seems like a weird thing to do in a lab, but okay.
Science is full of weird experiments. But when they did that, the enzyme absorbed light at a very specific wavelength, 450 nanometers.
Hence, P450, pigment 450.
Exactly. It's a spectral fingerprint. And they realized this pigment was a hemoprotein. It contains the same iron-containing compound found in hemoglobin in your blood.
Ah, so that's where the iron comes in.
Yes, and that iron is absolutely crucial for the chemistry it performs.
And is there just one P450? Or is it a team?
Oh no, it is a massive superfamily. In the human genome alone, there are at least 57 cytochrome P450 genes.
57. That seems like a lot of genetic real estate for one job.
It is. And they are incredibly versatile.
Huh.
The source text notes that they metabolize roughly half of all common drugs.
Half. That's huge.
It is. But they aren't just for drugs. They handle steroid hormones, carcinogens, pollutants. They even have normal physiological roles, like synthesizing steroid hormones or converting vitamin D into calcitriol.
So they are multitasking.
Constantly. They are the true Swiss Army knives of the liver.
Let's get into the chemistry for a second. The text gives a specific reaction equation. 3RH plus O2 plus NADPH plus H plus right RO, ROH plus H2O plus NADP plus long wall. Now, I can feel eyes glazing over in the audience. Can we break this down into a visual story?
Happily, let's visualize it in slow motion. RH is your xenobiotic, the drug or toxin, that's the target.
Okay.
It's sitting there in the enzyme's active site. O2 is molecular oxygen, the stuff we breathe. The P450 enzyme binds that oxygen.
Okay, so the P450 is holding the drug in one hand and an oxygen molecule in the other.
Right. But oxygen is stable. It doesn't want to react. You have to force it.
How?
You need energy. You need electrons. That's where NADPH comes in.
I remember NADPH from biology class. It's the battery pack.
It is. It carries high-energy electrons. But here's the tricky part. The P450 enzyme is a bit of a diva. It cannot accept electrons directly from NADPH. It needs a butler to hand them over. An enzyme called NADPH cytochrome P450 reductase.
That is a mouthful.
We can just call it the reductase. Its whole job is to take electrons from NADPH and shuttle them one by one to the P450.
It's like a bucket brigade for electrons.
Exactly. So the reductase passes an electron to the P450.
Yeah.
The P450 uses that charge to destabilize the oxygen molecule. Then, often a second electron comes in, sometimes from another helper called cytochrome B5.
And then what happens?
Crack. The P450 splits the oxygen molecule, O2 tears in half.
The oxygen split.
One atom of oxygen grabs two hydrogen ions and becomes water, H2O's. Harmless, it just floats away.
Okay.
But the other atom of oxygen, the P450, slams it right onto the drug.
Bam. And now the drug has a new handle.
It has a hydroxyl group, OH, and chemically that changes everything. It makes it reactive. It makes it ready for phase 2.
It's amazing to think that this controlled violence is happening on a molecular level millions of times a second in our bodies.
It is life at the atomic scale. Truly remarkable.
Now, you mentioned there are 57 genes. I see these codes in the text. CYP1A1, CYP3A4, CYP2D6. It looks like a license plate number or a library catalog code. How do we decode this?
It is a systematic nomenclature, and it's very logical once you know the rules. Let's break down CYP1A1.
Okay.
CYP just stands for cytochrome P450. That's the root. The first number 1 is the family.
And what defines a family? Are they related?
Yes. Members of the same family share more than 40% of their amino acid sequence identity. They are similar cousins.
Okay, so CYP1 is the family. Then there's a letter, A.
The letter is the subfamily. To be in the same subfamily, they must be very close relatives sharing more than 55% identity in their amino acid sequence.
And the final number.
That identifies the individual enzyme gene. So CYP1A1 is family 1, subfamily A, enzyme 1.
And there's a note here about italics. What's that about?
Yes, that's a key scientific convention. If you see CYP1A1 in italics, we are talking about the gene, the DNA code itself. If you see CYP1A1 in regular non-italic text, we are talking about the enzyme, the protein that's actually doing the work.
Gene versus enzyme. Got it. Where are these things located? I know we said liver, but are they just floating around in the soup?
No, they are anchored. They are embedded deep in the membrane of the smooth endoplasmic reticulum, SER.
Okay.
In liver cells, which we call hepatocytes and enterocytes, the cells lining your gut, they are incredibly abundant. In hepatic microsomes, these P450s can make up to 20% of the total protein.
20%. That's a huge amount of the cell's resources.
It shows you how critically important this defense system is. The liver dedicates a fifth of its microsomal protein just to this job.
Wow.
We also find some in the mitochondria, specifically in the adrenal glands, but those are mostly for making steroid hormones, not detoxing drugs.
Okay, so we have this massive army of enzymes, 57 different types, sitting in the liver membranes just waiting for chemicals. This leads us perfectly into Section 3, real-world implications. Because it seems like this system isn't perfectly neat and tidy.
It is anything but neat. It's messy. And that messiness is where medicine gets very, very complicated. The first concept we have to understand is overlapping substrate specificity.
That sounds fancy. What does it mean in simple terms?
It means the system is promiscuous. One specific P450 enzyme can metabolize many, many different drugs.
Okay.
And conversely, one single drug can be metabolized by several different P450 enzymes.
So it's not a simple lock and key situation where only one key fits one lock.
No, it's more like a master key system where some keys open lots of different doors, and this overlap is the root cause of almost all drug interactions.
Let's talk about that because the text brings up induction, this idea that the body adapts to what you put in it.
This is one of the most clinically dangerous and fascinating aspects of pharmacology. The liver learns.
Set the scene for us. Walk us through an example.
Okay, imagine you have a patient, let's call him Bob. Bob has a heart condition, maybe atrial fibrillation. He's at high risk for strokes, so his doctor puts him on warfarin.
Warfarin? That's a blood thinner, an anticoagulant.
Right. But it's a tricky drug. Give him too little. He forms a clot and has a stroke. Give him too much. He bleeds internally. The therapeutic window is razor thin.
So high stakes. The doctor finds the perfect dose, Bob's blood is just right, and he's safe.
Right. But then Bob develops seizures. So his neurologist prescribes a different drug, phenobarbital.
Now, phenobarbital is a generic sedative. It seems harmless enough in this context.
You'd think. But phenobarbital is a potent inducer. When Bob takes it, the drug travels to his liver. It binds to a receptor inside the nucleus of the liver cells and basically screams, "We need more enzymes! Build more factories!"
It cranks up the volume on the genes.
Specifically the gene for CYP2C9. Within a few days, Bob's liver cells have physically expanded. The smooth endoplasmic reticulum has hypertrophied. He has 3 or 4 times as much CYP2C9 enzyme as he did last week.
And CYP2C9 is the Pac-Man that eats warfarin.
Exactly. So suddenly, Bob's normal, carefully calibrated dose of warfarin is being chewed up instantly. It's gone before it can do its job. His blood gets thick again.
So he's at risk of a stroke again.
He is. So the doctor checks his blood, sees the warfarin isn't working, and says, "Okay, Bob, we need to double your dose."
And that works. For now, they double the dose, the hyperactive liver chews it up, and the balance is restored. Bob is safe. Again.
For now. Until Bob stops having seizures, and his neurologist tells him he can stop taking the phenobarbital.
Oh, no.
The signal to build more enzymes vanishes. Over the next week, the extra factories shut down. The CYP2C9 levels in his liver plummet back to normal.
But Bob is still taking that massive double dose of warfarin.
And now there are very few Pac-Mans to eat it. The warfarin levels in his blood skyrocket.
And Bob bleeds out.
It is a potential medical catastrophe, and it all comes down to gene transcription in the liver. That is why understanding induction isn't just academic. It saves lives.
That is an incredible and terrifying example. And it's not just about prescription drugs. Let's talk about alcohol and smoke. The double whammy.
Yes, this involves a specific enzyme called CYP2E1. This enzyme is known to metabolize industrial solvents and certain components of tobacco smoke. Okay. Many of these components in smoke are what we call procarcinogens. They aren't cancer-causing until they are metabolized by the liver.
So CYP2E1 is the enzyme that turns the smoke chemicals into cancer chemicals.
In many cases, yes. It performs that bioactivation step we talked about. Now, here's the kicker. Ethanol, the alcohol in drinks, induces CYP2E1.
So if you drink alcohol, you build more of the very enzyme that activates the carcinogens in smoke.
Correct. If you are a smoker who drinks, you are inducing the machinery that makes the smoke more dangerous. It significantly increases the risk of certain cancers compared to just smoking or just drinking.
It's a synergistic toxicity.
A perfect example of it.
That is a sobering fact, no pun intended. But let's flip it. We talked about induction, which is speeding things up. What about inhibition, slowing things down? The text mentions grapefruit.
The famous grapefruit effect. It's absolutely a real thing. Grapefruit juice contains compounds called furanocoumarins.
Furanocoumarins. Sounds exotic.
They are. And these compounds are potent inhibitors of cytochrome P450. They don't make the liver build more enzymes. They break the ones you already have. They basically gum up the works. They block the enzyme's active site or cause the protein to be destroyed.
So if you take a drug like a statin for cholesterol or omeprazole for acid reflux and you drink grapefruit juice,
the enzyme that is supposed to break down that drug is blocked. The drug isn't broken down. It builds up in your blood to potentially toxic levels.
So you essentially overdose on your normal prescription.
Yes, just from your breakfast juice. And interestingly, if the drug is a prodrug, one that needs the enzyme to start working, grapefruit juice would have the opposite effect.
It would stop the drug from working.
Entirely, because the activation step is blocked.
It's amazing how much diet influences this. But it's not just diet, is it? It's genetics, too. The text talks about polymorphism.
Yes, this is a huge field. Polymorphism just means that within the human population, there are different versions or alleles of these genes. This explains why you and I might take the same Tylenol and have very different reactions.
The text mentions a fascinating case. A smoker's gene, CYP2A6.
This is one of my favorite examples of how genetics can influence behavior. CYP2A6 is the main enzyme responsible for metabolizing nicotine into cotinine. It breaks nicotine down and clears it from the body.
So if you have a really fast, efficient CYP2A6, you burn through your nicotine buzz quickly.
Exactly. And what does a smoker do when the buzz wears off?
They light another cigarette.
Right. But some people have what are called null alleles. Their version of the CYP2A6 gene is broken. The enzyme is inactive.
So they metabolize nicotine very, very slowly.
Correct. The nicotine stays in their blood and their brain for a much longer time.
And the behavioral result of that is?
They smoke less. They are statistically much less likely to become tobacco dependent because they don't get those cravings and withdrawals as quickly or as often.
That's incredible. The text suggests we could even use this for smoking cessation.
Right. The idea is, if we could invent a drug that inhibits CYP2A6, essentially making a normal person's enzyme act like the null allele, we could help people smoke less because their nicotine levels would remain stable for longer.
That is the definition of precision medicine.
It is. It's using genetics to guide therapy.
Okay, we've survived phase 1. We've oxidized the molecule. We've maybe created a monster in the process. Now we need to fix it. Let's move to section 4. Phase 2, conjugation and excretion.
Right. Phase 2 is the cleanup crew. The goal, remember, is to make things polar or water-soluble. We have this reactive handle from phase 1, and now we need to attach something huge and water-loving to it to tag it for excretion.
What's the most common way the body does this? The main pathway.
Glucuronidation.
That is a mouthful.
It is. It involves attaching a molecule of glucuronic acid to the xenobiotic.
Where do we get glucuronic acid? Is that something we eat?
It comes from glucose. The body makes it. It's basically a sugar molecule that's been modified to be an acid. The actual donor molecule is called UDP glucuronic acid, and the enzymes that do the pasting are called glucuronidyltransferases.
And what kinds of things get targeted by this?
Oh, a huge list. The text mentions 2-acetylaminoflurane, which is a carcinogen, aniline, benzoic acid, probamate, which is a tranquilizer, phenols, and many of our own steroid hormones.
So the body just plasters this sugar acid onto them.
Yes. It can attach to oxygen, nitrogen, or sulfur groups on the molecule. Think of it like putting a giant, wet, heavy backpack on the toxin. Once that glucuronide tag is on, the molecule is bulky and charged. It cannot slip back into the fat. It is definitely going out in the urine or bile.
What are the other options besides glucuronidation? What if that pathway is busy?
Sulfation is another big one. The body attaches a sulfate group.
Like sulfur?
Yes. The donor here is a really cool molecule called PAPS, adenosine 3-phosphate phosphosulfate. We just call it active sulfate. This is particularly used for alcohols, arylamines, and phenols.
And then there's acetylation.
Acetylation is crucial because it has a very famous genetic component that affects drug therapy. The enzyme uses acetyl-CoA, which we have tons of from metabolism, to tag the drug.
The example given is isoniazid.
Right, a frontline drug for treating tuberculosis. It's metabolized by acetylation. But here's the thing. People are genetically split into two groups, slow acetylators and fast acetylators.
I'm guessing this is just like the nicotine thing with CYP2A6.
Exactly the same principle. If you are a slow acetylator, you can't clear the isoniazid quickly. It builds up in your blood.
Which means you're more likely to suffer toxic side effects.
Precisely. Specifically nerve damage in the case of isoniazid. If you are a fast acetylator, you might clear it too fast and the treatment might not work as well unless the dose is adjusted.
It really emphasizes that one dose fits all is a complete myth.
It absolutely is. And for some drugs, knowing a patient's acetylation status can be the difference between a cure and toxicity.
I want to move to section 5 because there's one molecule that seems to be the unsung hero of detoxification. Glutathione.
Ah, GSH. The body's defense shield. If P450 is the sword that creates danger, glutathione is the shield that absorbs it.
What is it exactly? It's not a protein, right?
It's a tripeptide. That means it's made of just 3 amino acids linked together: glutamate, cysteine, and glycine. The full chemical name is gamma-glutamylcysteine.
I'll stick with glutathione. What does it do?
It is the master conjugator for electrophilic compounds. Remember those reactive, dangerous intermediates that phase 1 created? The ones looking to steal electrons and damage DNA.
The bomb.
Glutathione throws itself on the bomb. An enzyme called glutathione S-transferase, GST, catalyzes a reaction where the R group, the toxin, is attached to the sulfur atom of glutathione.
The text shows the reaction. $Ra plus GSH, RIDAR, RSG.
Exactly. This neutralizes the electrophile. The sulfur atom in that central cysteine amino acid is the magic bullet here. It forms a very stable bond. The text mentions something really interesting about these GST enzymes. They have a ligandin function. What does that mean?
Yes. This is fascinating. Sometimes they don't even metabolize the toxin immediately. They just bear hug it.
A bear hug.
They bind non-covalently to things like bilirubin, steroid hormones, or carcinogens just to hold them. They transport them or sequester them to keep them from bouncing around the cell and damaging critical things like DNA. It's a physical defense as much as a chemical one.
But there's a downside here too, right? The text says GSTs are upregulated in tumors.
This is the tragic irony of cancer biology. Cancer cells are cells too, and they want to survive. So cancer cells often produce massive amounts of glutathione S-transferase.
So when we try to poison the cancer with chemotherapy,
the cancer cell uses its extra GSTs to conjugate the chemotherapy drugs and pump them out of the cell. It creates drug resistance. The cancer protects itself using the very system that's meant to protect us.
that is diabolical.
It's evolution in a petri dish.
So glutathione gets used up in this process. It sacrifices itself. What happens to the glutathione toxin conjugate?
It enters a pathway called the mercapturic acid pathway. The conjugate travels to the kidney. There it gets chopped up a bit. The glutamate and glycine are removed, leaving just the cysteine attached to the toxin.
Okay.
Then it gets acetylated. The final product is a mercapturic acid, which is then easily peed out.
Glutathione isn't just for detox though, right? It has other day jobs.
Oh, absolutely vital ones. The text lists several. 1, it reduces hydrogen peroxide to water via an enzyme called glutathione peroxidase. This protects us from oxidative stress.
Important.
2, it acts as a general intracellular reductant. It keeps the sulfhydryl, the DKSH groups of other enzymes in the correct reduced state so they can work properly.
Okay. And the third one was weird. It helps transport amino acids.
Yes, across cell membranes, especially in the kidney and liver. This involves a pathway called the gamma-glutamyl cycle.
And there's a clinical diagnostic tip here regarding an enzyme called GGT.
Gamma-glutamyltransferase, or GGT. This enzyme is part of that amino acid transport cycle. It lives on the plasma membrane of liver cells. If the liver is damaged from alcohol or a blocked bile duct, for instance, the membranes get leaky and GGT spills out into the blood.
So if a doctor sees high GGT levels in a blood test,
It's an early marker of liver trouble. It's often one of the first signs of alcohol abuse or biliary obstruction. It's a big red flag waving in the bloodstream.
fascinating. It's also interconnected. Okay, section 6. We've talked about how the system works. But sometimes the xenobiotics win. Let's talk about toxicity. The text categorizes the damage into 3 main headings.
Right. Cytotoxicity, which is cell injury or death, immunological effects, and carcinogenesis, which is cancer.
Let's start with cytotoxicity. How does a chemical actually kill the cell? What's the mechanism?
It usually starts with covalent binding. The reactive metabolite that phase 1 created permanently binds to DNA, RNA, or essential proteins. It just gums up the machinery of the cell.
And the cell notices this damage. It tries to fight back.
It does. Specifically, if it damages DNA, the cell panics. It activates repair mechanisms. One of these is a key enzyme called poly-ADP ribose polymerase, or PRL.
PRP tries to fix the DNA.
It acts like a first responder at a disaster site, but to do its job, it consumes a huge amount of NAD.
NAD is crucial for energy metabolism, right?
Yeah.
For making ATP.
It's the fuel for ATP production, so massive DNA damage leads to massive PRP activation, which leads to massive NAD consumption.
The cell runs out of gas.
The cell literally runs out of energy. ATP levels crash. The cell can't maintain its membranes or do anything else. It dies. That is cytotoxicity. It's an energy crisis triggered by DNA damage.
Wow, okay. Second heading, immunological effects. This is where the body starts attacking itself.
Yes. This relates to a concept called haptin. Sometimes a xenobiotic is too small for the immune system to notice. It flies under the radar. But if it covalently binds to one of the body's own proteins, it changes that protein's shape.
It puts a funny hat on the protein.
Exactly. And the immune system looks at this new complex and says, I don't recognize you. You are an invader. attack.
So it creates antibodies against the modified protein.
Right. But here's the problem. Those antibodies might also start attacking the unmodified, normal, healthy protein because they look similar enough.
And now you have an autoimmune disease.
Exactly. The drug has triggered a civil war inside your body.
Finally, the big one, carcinogenesis, cancer. We touched on this with the smoke, but walk us through this specific mechanism mentioned, epoxides.
Right. Some chemicals like benzoaperine, which you find in grilled meat and tobacco smoke, are indirect carcinogens. They need P450 to activate them. When P450 adds that oxygen atom, it can create a structure called an epoxide.
Chemically, what is an epoxide? What makes it so bad?
It's a little triangular ring of 2 carbons and an oxygen. It is highly strained, chemically unstable, and highly reactive. It's like a loaded mousetrap.
Okay.
It desperately wants to break open and bind to something, and that something is often the nitrogen atoms in your DNA bases.
Right.
That causes mutations.
Do we have a defense against epoxides?
We do. We have a specific enzyme called epoxide hydrolase.
And what does that do?
It takes that dangerous strained epoxide ring and adds water to it. It pops the ring open safely, turning it into a much less reactive dihydrodyl.
Which is safer?
Much, much safer. So your risk of cancer from a chemical like this often comes down to a race.
A race.
A race between cytochrome P450, which is making the epoxide bomb, and epoxide hydrolase, which is diffusing it.
And because of genetics, some people have faster bomb makers and some have slower bomb diffusers.
Precisely. That balance encoded in your genes can determine your susceptibility to chemical carcinogens.
Wow. It really puts everything in perspective. So let's wrap this up. We've gone from the definition of xenobiotics to the dangerous phase 1 activation, the P450 superfamily, the induction wars between drugs, the phase 2 cleanup with glutathione, and finally, how it all can go so wrong with cancer and cell death.
It is a complex, elegant, and sometimes very dangerous dance happening in your liver right now as we speak.
What's the big takeaway for you, if you had to summarize this entire chapter for the listener?
I think the takeaway is that detox isn't a juice cleanse you buy at the store. It isn't a foot pad you put on at night. It's a sophisticated, powerful, enzymatic process encoded in your DNA. It involves phase 1 functionalization and phase 2 conjugation. And it is heavily influenced by your genetics, your diet watch the grapefruit, and your environment.
And my takeaway is the double-edged sword. The fact that the very enzymes designed to protect us, the P450s, are the ones that can turn a harmless chemical into a deadly carcinogen. We are walking a chemical tightrope every single time we eat or breathe.
We are. And understanding that tightrope is the key to modern medicine and toxicology.
Here's a final provocative thought for you to chew on, listener. We talked about biochemical individuality, how your unique mix of CYP enzymes determines your reaction to coffee, drugs, and smoke. But think about this. If we continue to pollute our environment with new synthetic xenobiotics that didn't exist in nature 100 years ago, are we outpacing our own evolution? Can our P450s, which evolved to handle plant toxins and smoke from fires, keep up with the chemical cocktail we are creating, or are we driving the evolution of our own toxicity?
That is the question of the century.
Thanks for diving deep with us. We'll see you next time.